Field of analytical chemistry, specialized apparatus is fundamental for accurate analysis, quality control, and research. Industries rely on a variety of sophisticated instruments to identify, quantify, and study chemical substance, ensuring product integrity and compliance with standards. Key apparatus in analytical chemistry includes spectrometers, chromatograph titrators, and electrochemical analyzer. Spectrometers used for identifying and quantifying molecules based on their interaction with light. Chromatograph essential for separating and analyzing complex mixture. Titrators used for precise measurement of chemical concentration. An electrochemical analyzer is important for detecting and measuring ions and other analytes in solution. Several industries rely heavily on analytical apparatus for various applications. One of the industry is microbiology industry. The microbiology industry uses specialized equipment to advance research, diagnostics, and production. Key tools include microscope for observing microorganisms, incubators for growing culture and for sterilizing equipment, and autoclave PCR machine for DNA amplification. Advanced technology like automation and digital imaging enhances accuracy and efficiency. These tools are crucial for detecting pathogens, improving workflows, and ensuring quality in products like vaccine and antibiotic. Understanding these apparatus is essential for scientific and industrial progress.
Now I will explain about the theory on the application. The first one is the analytical balance. The scientific contribution, which is the concept of precise weighing, dates back centuries. But modern analytical balance owe their accuracy to advancements in electromagnetism and precision engineering. There are researchers that include Joseph Louis Galuzek, who developed the principle of buoyancy, and Thomas Graham, who studied diffusion and osmosis. And their work led to the groundwork for precise measurements in chemistry and microbiology. The second one is the autoclave. In scientific contribution, the autoclave's effectiveness in sterilization relies on the principle of heat and pressure. It owes its existence to the pioneering work of Charles Chamberlain and Charles Edward Brown-Séquard, and the researchers, which are Charles Chamberlain, a French bacteriologist, collaborated with Louis Pasteur and developed the first practical autoclave. And his work significantly improved laboratory hygiene and also safety. The third one is the incubator. The scientific contribution of the incubator, which is the concept of controlled incubation for microbial growth, emerged from studies on fermentation and the bacterial cultures. There are several researchers that can be included in this. The first one is Louis Pasteur, a giant in microbiology, contributed significantly, in which his experiments on fermentation and pasteurization led to the development of incubator. Additionally, Robert Koch, known for his work on tuberculosis and anthrax, refined incubation techniques for isolating bacteria. Lastly, I will be talking about the theory on the other apparatus. The first one is the Bunsen burner. It is named after Robert Bunsen, and this gas burner revolutionized laboratory heating. Bunsen collaborated with Gustav Kirchhoff to develop it. Next, we have the centrifuge. The centrifuge principle stem from the work of the Swedish chemist Theodor Svedberg, who studied colloids and sedimentation. Next is microscope. Anthony van Leeuwenhoek, a Dutch scientist, pioneered microscopy and observed the microorganisms using the simple lenses. And lastly, the pH meter. In this, Fritz Haber and Zygmunt Clemencevic contributed to the development of pH measurement techniques.
Next, I will explain about the application and the uses of the apparatus that were needed to prepare the culture media experiment, which is preparing the agar plates. There are three apparatus that were used while preparing the agar plates. First of all, hot plate. Hot plate was used to boil the agar mixture. To ensure proper dissolution and sterilization of the media during the preparation of agar, the mixture must be heated to a specific temperature. Secondly, the analytical balance. Analytical balance plays a critical role in preparation and measurement of agar. Agar plates require precise measurements of agar powder or flakes, as well as other ingredients such as nutritive, salts, and additives. Lastly, the ovens. Ovens used for sterilization the petri dishes before and after the experiment. The reason the plate is inverted is the fact that the airspace between the dish lid and the agar surface is saturated with moisture. During the incubation, the moisture condenses on the upper lid as droplets. Now I'm going to explain about the methodology of preparation of agar mixture. First, heat a beaker and petri dishes in a 177 degrees Celsius oven for 2 to 3 hours. Second, prepare the agar medium using nutrient agar powder. Pour 180 ml plus 22 ml of distilled water into the beaker along with 4.6 gram of agar powder measured using analytical balance. Next, cool down the beaker and pour the medium ingredients into the beaker. Fourthly, turn a hot plate to high and set the beaker on it. Bring the mixture to boil on hot plate for 1 minute. After that, turn off the heat and leave the agar medium to cool for a few minutes until it reaches 49 degrees Celsius.
Polymerase chain reaction (PCR) machine. The application for this machine is to amplifying DNA sequences. For example, PCR machines enable researchers to amplify specific DNA regions from microbial samples, making it possible to study genetic material even from small or degraded samples. PCR is used to detect pathogens, identify genes, and investigate genetic variations. For instance, PCR is crucial for diagnosing infectious diseases by amplifying pathogen-specific DNA sequences. Gel electrophoresis apparatus. This apparatus is used to separating DNA or protein fragments. For example, gel electrophoresis separates DNA, RNA, or proteins based on size and charging. Researchers use this technique to analyze PCR products, check the quality of extracted nucleic acids, or study protein expression. For example, after PCR, gel electrophoresis can confirm the presence of size of amplified DNA fragments. Colony counter. This machine is used to counting bacterial or fungal colonies on agar plates. For example, colony counters help researchers quantify microbial populations by counting the number of colonies formed on agar plates. This is used to determine the concentration of microorganisms in a sample, evaluate the efficacy of antimicrobial agents, or monitor contamination levels. For instance, after spreading a diluted sample on an agar plate, researchers count the colonies to estimate the original concentration of bacteria. Bioreactor. This machine is used to cultivating microorganisms under controlled conditions for large-scale production. For example, bioreactors provide a controlled environment for growing large volumes of microorganisms, optimizing conditions for maximum yield. Researchers use bioreactors to produce antibiotics, enzymes, biofuels, and other microbial products on an industrial scale. For example, bioreactors are essential for producing penicillin, where the growth conditions of Penicillium molds are carefully regulated to maximize antibiotic production. As the conclusion, the use of specialized apparatus in the microbiology industry is critical for advancing research, ensuring precision, and maintaining sterile conditions essential for successful experimentation and production. From the sterilization capabilities of autoclaves to the precise amplification of DNA using PCR machines, these tools enable researchers to explore and harness the potential of microorganisms effectively. Incubators, microscopes, centrifuges, and other apparatus facilitate the study of microbial growth, morphology, and genetic material, contributing to the identification and characterization of various species. Additionally, bioreactors and other large-scale equipment support the industrial production of valuable microbial products such as antibiotics and enzymes. Overall, the integration of advanced apparatus in microbiological research and industry not only enhances our understanding of microorganisms but also drives innovation and efficiency in biotechnological applications, leading to significant scientific and economic benefits.